Welcome to the Innate Immunity module, and in this talk we're going to focus on the lectin pathway. We will learn about the lectin pathway, and in that we will focus on how the pathway gets activated and what is the outcome of that pathway. The complement proteins are a collection of more than 30 different soluble proteins that are present in blood as well as bodily fluids. They were initially discovered in vertebrates. However, now we know that homologs of some complement proteins are also found in invertebrate organisms. A lot of complement proteins are thioester proteins, and when we look at vertebrates, the complement proteins are mainly synthesized in the liver. Now, the complement proteins work together and form a cascade that's called as the complement pathway. The complement pathway aids in the pathogen elimination as well as induction of inflammation. These complement proteins on their own are not antimicrobial in nature, but when the complement pathway gets activated, that is when pathogens can get eliminated. Now, when a pathogen is able to overcome the epithelial barriers of the host and enter the host, that is when the complement system is activated. There are three main pathways of complement activation that have been studied in vertebrates. The first one is called as the alternative pathway. The second one is the lectin pathway. And then we have the classical pathway. Now, the complement system is an ancient form of host defense. And so when we look at the evolution of the complement pathways, the alternative pathway evolved first, followed by the lectin pathway, and then the classical pathway. The lectin pathway is activated by complement proteins that have the ability to recognize and bind to carbohydrate moieties that are found on microbial surfaces but not on host cells. So let's briefly look at different carbohydrate moieties that are found on microbes and on host cells. So in this figure, when we look at the first panel that's shown in black, we are looking at the basic carbohydrate moiety that is normally added to proteins in the ER. In this moiety, we can see that there are glucose, mannose, and N-acetylglucosamine residues. Now, when we look at yeast, which is a type of fungus and can cause infections, there is some alteration and addition of added sugars to this basic carbohydrate moiety. When we look at what kind of sugars are there towards the end of that carbohydrate chain, we can see that it is very rich in mannose. Now, on the other hand, when we look at the carbohydrate moieties present on vertebrates like us, when we look at the ends of those carbohydrate chains, we don't see mannose the way we saw in yeast. On the other hand, we see that there are sugars like sialic acid and galactose and also fucose that are there towards the ends of those carbohydrate chains. And thus there is a difference in the carbohydrate moieties that are present on proteins in microorganisms and on vertebrates like us. The lectin pathway is a pathway that involves the activation of complement proteins like C4 and C2. When these complement proteins get activated, they associate to form an enzyme called the C3 convertase. This C3 convertase then activates the complement protein called C3, which then is able to associate with the C3 convertase to form another enzyme called as the C5 convertase. The C5 convertase is then able to bind to the complement protein C5 and is able to cleave the complement protein C5. One of the cleavage products of C5 is able to initiate the terminal part of the lectin pathway. So let us look at this entire process in more depth. The lectin pathway is activated when proteins are able to bind to carbohydrate moieties that are found on the surfaces of microorganisms. The complement proteins that are able to identify these kinds of carbohydrate moieties are Mannose Binding Lectin and Ficolin. they both are able to identify different types of carbohydrate moieties. And let us first look at the Mannose Binding Lectin. Mannose Binding Lectin is a protein that has a lectin domain, which is also called as a carbohydrate recognition domain, or CRD, followed by an alpha helical coiled coil domain and a collagen helix. Now, 3 MBO monomers can come together to form a trimer. And in this trimer, we can see that the carbohydrate recognition domains of all 3 molecules are close to one another. These trimeric clusters can come together to form oligomers. In the case of this oligomeric structure, the carbohydrate recognition domain, or the lectin domain, has the ability to bind to mannose and fucose residues that are present on the cell surface of an organism, but not to sialic acids. Mannose is known to be present on the surface of many microorganisms, especially fungi, and so the Mannose binding lectin is able to recognize these microorganisms. Ficolins are proteins that are also able to activate the lectin pathway. When we look at the structure of ficolin, it is very similar to that of the Mannose binding lectin. Now, ficolins have a carbohydrate recognition domain that is able to bind to oligosaccharides that contain acetylated sugars, but not mannose-containing carbohydrates. Examples of organisms that have acetylated sugars as part of their cell surface are bacteria, because if you remember, peptidoglycan is made up of N-acetyl glucosamine and N-acetyl muramic acid, and thus they're acetylated sugars. When we look at the structure of the ficolin, here we can see that it also can form a trimeric structure where we have the fibronectin domains together, and these trimeric structures can come together to form an oligomeric structure, and those fibronectin domains will be able to bind to these acetylated sugars present on the microbial surface. The different types of ficolins that are present are L-ficolin, M-ficolin, and H-ficolin. MBL and ficolins are associated with proteases called as MBL-associated serine proteases, or MASPs. In the presence of a pathogen, MBL or ficolin will bind to carbohydrate moieties present on that pathogen's surface, and this results in the activation of MASP1. MASP1 can then activate MASP2, and this way the MASP proteases become active. The MASP proteases then can cleave 2 complement proteins called C4 and C2. MASP2 is able to cleave C4 to form 2 fragments. The smaller fragment is called C4a and the larger fragment is called C4b. Now, C4 is a thioester protein, and when C4b is produced, it has a reactive thioester bond. Now, this thioester bond can react with hydroxyl or amino groups that are present on the cell membrane of that pathogen. And due to this reaction, a covalent bond is formed between that thioester and the hydroxyl or amino group. And this is what allows C4b to covalently bind to the surface of the cell. Now, this reaction has to happen very quickly, and if this reaction does not happen quickly, then the bond is hydrolyzed, so the bond will react with the surrounding water, and then no binding will occur to the cell surface because the thioester bond has been hydrolyzed. Hence, the process has to happen very quick, or else no covalent bond will be formed between proteins that are on the surface of the cell and C4b. Thus, just to remind everybody, C4b that is generated from the cleavage of C4 is able to bind to the surface of the pathogen. In addition to cleaving C4 to form C4a and C4b, MASP2 can also cleave C2 into two fragments. The smaller fragment is called C2b, which is an exception to the rule, because in the complement nomenclature, normally the larger fragment is associated with the alphabet B. So just a reminder, C2 is an exception to the rule, and hence the smaller fragment is called C2b, while the larger fragment is called C2a. Now, the C4b and C2a that have been generated through the cleavage of C4 and C2 associate with one another to form a complex called C4b2a. And this C4b2a complex has enzymatic activity, and it's called as the C3 convertase. And what the C3 convertase does is that it can cleave the molecule C3, which is another complement protein, and it cleaves it into two fragments. One of them is called C3a, which is the smaller fragment, and the other one is called C3b, which is the larger fragment. Now C3, which is cleaved by the C4b-C2a complex, which is also called as the C3 convertase, it generates a smaller fragment called as C3a, which is a soluble molecule. So the C3a does not bind to the surface of the pathogen, but on the other hand, it can diffuse because it's soluble, and it is an anaphylatoxin. An anaphylatoxin is a molecule that is able to promote acute inflammation. Now, C3a is able to promote acute inflammation by enabling chemotaxis to occur, increasing permeability of blood vessels, and activating immune cells. The C3b fragment that is formed, on the other hand, is deposited on the pathogen surface because the C3b molecule has a reactive thioester bond similar to the C4b molecule. And this reactive thioester can covalently bind to hydroxyl or amino groups that can be present on the surface of the pathogen. Thus, the pathogen surface is coated by C3b, and C3b functions as an opsonin. And opsonin is an agent that promotes opsonization, which is another name for phagocytosis. So immune cells that have complement receptors that recognize C3b can phagocytose the pathogen that is coated with C3b, and thus it can destroy the pathogen. The C3b that is formed can also bind to C4b2a, which is the C3 convertase, to form another complex called C4b2a3b. And this complex also has enzymatic activity and is called as the C5 convertase. This C5 convertase now has the ability to cleave the complement protein C5 into two different fragments. One of the fragment that is generated, which is the smaller fragment, is C5a. Now C5a, like C3a, is a soluble and diffusible protein fragment, and it functions as an anaphylatoxin. The anaphylatoxin is able to promote acute inflammation by increasing the permeability of blood vessels, by also promoting chemotaxis of immune cells to the site of infection, and by activating some immune cells. The other fragment that is generated by the cleavage of C5 is C5b. And C5b is able to initiate the late or terminal events of complement activation, which is the formation of a membrane attack complex, which forms a pore-like structure on the phospholipid bilayer of the pathogen, and thereby it promotes cell lysis and killing of the pathogen. Now let us see how the membrane attack complex is formed. Once C5b is generated, it is able to bind to another complement protein called C6. The complex of C5b6 is able to recruit another complement protein called C7, And the interaction of C7 with C5b6 is able to expose a hydrophobic region on C7. And this allows the C5b6-7 complex to be on the plasma membrane of the pathogen. They recruit another complement protein called C8, and thus we form the complex of C5b, 6, 7, and 8. This complex can recruit another complement protein called C9 to form another complex. But the neat thing is it doesn't recruit only one C9 molecule. It can recruit many C9 molecules, anywhere from 10 to 16 of them. And these C9 molecules arrange themselves in a ring structure to form the membrane attack complex. The membrane attack complex forms a channel-like structure in that phospholipid bilayer of the pathogen. And this pore-like structure is able to disrupt the proton gradient across the membrane of the cell because it allows the flow of different ions and other substances between the cell and the environment. The membrane attack complex also allows water and enzymes like lysozyme to enter the cell from the environment, and thus the cellular integrity is compromised, which results in the death of that cell. With this, we come to the end of the lectin pathway. So to summarize this pathway, the lectin pathway is activated when complement proteins like MBL and ficolin are able to recognize and bind to carbohydrate moieties that are present on pathogen cell surfaces. This binding activates proteases that are associated with MBL and ficolin called MASPs. The activated MASPs are able to cleave two complement proteins called C4 and C2. These two complement proteins are cleaved into two fragments that are designated as A and B. The C4b fragment is able to interact and covalently bind with the surface of the pathogen, and it's able to associate with the C2a fragment to form the complex C4b2a. This C4b2a complex has enzymatic activity and is called as the C3 convertase. The C3 convertase is able to bind to C3 and cleave it into two fragments. The smaller fragment, which is called C3a, is an anaphylatoxin and can promote inflammation. It is also soluble and hence it can diffuse from the site of production. The larger fragment is C3b. C3b is also able to form a covalent bond with the surface of the pathogen, and hence it is able to coat the surface of that cell. It functions as an opsonin because it facilitates the uptake of the pathogen by immune cells through the process of phagocytosis, which is also called as opsonization. The C3b additionally can also interact with the C4b2a complex to form the complex C4b2a3b, and this is called as the C5 convertase. The C5 convertase is able to interact with the C5 protein, and the C5 complement protein is then cleaved into two fragments, which is the smaller C5a fragment, which is a diffusible molecule and functions as a potent anaphylatoxin that promotes inflammation, and a larger C5b fragment that is able to interact and recruit with other complement proteins like C6, C7, C8, and C9. They will form the membrane attack complex, which will result in cell death of that pathogen. Thus, we come to the end of this talk where we learned about the lectin pathway, and we also learned how the lectin pathway can cause the death of the pathogen and induce inflammation through the production of anaphylatoxins.